Hey everyone, I'm going to go through our next stage, which is called pyruvate oxidation and the citric acid cycle. So this is going to be the phase after glycolysis, and this is going to occur in our mitochondrial matrix. So again, here's that recap. As we talked about glycolysis, we know that sugar splitting, it's going to go through a net release of 2 ATP. So that's this little molecule down here. 2 NADH is going to be released, and a water molecule as well is going to be released during glycolysis. And the whole main point is taking that glucose and splitting it from that 6-carbon sugar to break it down into 2, 3-carbon pyruvate molecules. And those pyruvate molecules will then be used for our next step, pyruvate oxidation, which will transfer those into a new molecule to enter into the citric acid cycle and allow for a net of 2 ATP in the end. So again, glycolysis was outside of the cell in the cytosol, and then the rest of our cell respiration process is all happening inside the mitochondria, the powerhouse of the cell, or the inside its mitochondrial matrix. So again, here's our structure. A transport protein does have to pass those pyruvate molecules through the mitochondria to enter in here. And we see this complex folding pattern of the matrix, and then this extra folding in surface area, as we learned about before, that allows for more complex and efficient chemical reactions to occur. So that citric acid cycle is more complex, so we can have a lot of chemical reactions happening in here that are controlled. So this is the general overview that we should kind of understand by the end of this process for our 2 steps. Pyruvate oxidation is often referenced as a link phase. So you'll see that in your POGL when we do that as kind of a review. It's called the link phase or pyruvate oxidation. And then we have the citric acid cycle, which looks a lot like, the Calvin cycle for photosynthesis, right? So going back here to glycolysis, that's happening outside in the cytosol. Remember, glucose is a 6-carbon sugar. It's broken down into pyruvate, which are 2, 3-carbon molecules here, as we can see with our gray spheres. Transport protein will carry those pyruvate molecules into the mitochondrial matrix. During this step, so we've got pyruvate into the mitochondrial matrix. There'll be an enzyme complex that is going to remove this carbon dioxide over here. So carbon dioxide out as you breathe and are watching this video. Carbon dioxide is leaving through pyruvate oxidation here. It will then convert over here on the left side. It'll strip away those electrons and convert NAD plus to NADH. It's going to add this coenzyme A to our remaining molecule. So coenzyme A enters in, and that creates what is known as an acetyl-CoA molecule. And that's the important molecule that then can run through the citric acid cycle on this enzymatic pathway that we see down here. There will be 2 acetyl-CoA molecules for each glucose that we have, right? So we had originally 2 3C molecules. This is just representing 1 pyruvate converting into 1 acetyl-CoA, but remember there's going to be 2 that enter through here. So just make sure we're on the same page. Again, this is happening in the matrix of our mitochondria, and the whole job is to complete the breakdown of glucose, right? So we have carbon dioxide released over here already, but as it goes through the citric acid cycle, it's going to complete that process. And so we can see 2 carbon dioxide molecules will be removed again and released as waste. Let's see. And then continuing through here, each turn we'll have 2 carbon dioxide molecules go through. We'll have 3 NADH molecules released. We'll have, I'll let my little blinker move away. We'll have 1 ATP released over here. and then we'll have one FADH released over here. So that's just one turn. Remember, we have a second acetyl-CoA go through, so we're actually going to have a net result of 4 carbon dioxide molecules out, 6 NADH molecules released, 2 ATP, and 2 FADH molecules at the end. So again, looking at this whole process, the original 6-carbon sugar that occurred at the beginning of glycolysis, that has gone through, and some of those carbons were released as carbon dioxide, right? As we are just breathing here. Only 2 ATP molecules are released during this process. Where is all the other energy, you might be wondering? That is found and stored in the bonds of our NADH molecules and our FADH molecules. So we have 6 NADHs and we have 2 FADHs that have all that energy stored. Those electron carriers are going to move on to the electron transport chain, which will go in our next concept, but those are going to be really important electron molecules. Again, so if we look at the process of pyruvate oxidation, to give you a more focused view of it, pyruvate enters as a 3C molecule. Carbon dioxide is produced and NADH is produced. It's converted into acetyl-CoA, which is also going to be used in that citric acid cycle or the Krebs cycle here. And so we'll have our net gain of 2 ATP by the very end of it. But really, our acetyl-CoA molecules, they get converted into this thing called citrate, which I'll show you in a moment, and carbon dioxide. And this is just kind of a repeat of what I was saying before. Remember, we talked about phosphorylation happening at a substrate level. So an enzyme is forcing ADP and our PI molecules to interact with each other. So that's how we get our ATP net gain. This is at the very most basic level. This is what we should know about the citric acid cycle. This is truly what's happening, right? This cascade of enzymatic processes, but you don't have to memorize these steps. In goes our acetyl-CoA molecules, or that pyruvate that lost to carbon dioxide that's going to enter in over here. And we can see that's where it gets converted to this molecule called citrate, and that's where citric acid cycle gets its name. It's also called Krebs cycle because, a guy named Krebs named it after himself, but it already had the name of citric acid cycle and didn't really take off that way. So we can see the cycling of this molecule, and basically we can see the gain and loss of carbon atoms all the way throughout this cycle, right? And that's really what we focus on. But we can see the release of NADH molecules and FADH and ATP throughout this. And remember, there's 2 acetyl-CoA molecules that actually enter in. So that's where we get our 6 NADH molecules, our 2 FADH molecules, and our 2 ATP. And here's our summary. All right. And we do have the output over here, the additional NADH molecules from our intermediate step or the link phase from earlier on. So that's where the 8 NADH molecules come from.